So now that we've described everything in details about making a genomic DNA library and the steps that are involved in genome sequencing, let's have a closer look at genome sequencing and the problems that may arise and how do you troubleshoot those problems in genome sequencing. So here is a representation of the whole genome shotgun approach. This is your genomic DNA, and the top strand is represented here as 5' end and 3' end. And these are your individual fragments that were cloned and sequenced. And this represents the unique overlaps that are found between two adjacent fragments. So therefore, now that you have unique overlap between 1 and 2, so you know 2 follows 1, likewise 3 follows 2. Likewise, 5 is followed by 6, is followed by 7, is followed by 8. And this unique overlaps are determined after you got the sequence of individual fragments and you compare the 5' end of 1 with 3' end of all the rest of the fragments, right? And here, this one is a representation of the fragment that is not taken into sequencing. Although a library is complete, when you're randomly picking up the fragments, you did not pick up this fragment here for sequencing, right? And so therefore, what you can do is you could join all the sequences that you've got in continuation together, forming what is known as a larger sequence collection, that is your Contig 1 here. Likewise, since 5, 6, 7, 8 are in continuation, you can put their sequence together and represent the overlap only once, giving you what is known as the Contig 2 here. And because you randomly picked up fragments of sequencing and you did not pick up the fragment 4 here, the Contig 1 cannot directly be joined to Contig 2. So this represents a gap in the sequence, right? And we'll talk more about it as we go along. So here you are, your steps in sequencing. First, of course, for sequencing, the library is spread on a culture plate, incubated, and individual colonies are sequenced. A few colonies are picked up at random and sequenced. So these sequences are then compared with respect to their ends, and you're comparing the 3' end of one fragment with the 5' end of all other fragments to check for which two fragments have unique overlaps and allow you to order them together. And as long as you keep getting continuous overlaps, you put all the sequences together into what is known as a Contig or contiguous sequence or continuous sequence. So all sequences with unique overlaps and in continuation are clubbed together into Contigs. As I said, you could have what is known as a gap. So let's now talk about gaps. This is your genomic DNA again, and only the top strand is represented. So you have the 5' end here and the 3' end here. And then, of course, you have your complete genomic DNA library. But during the time of sequencing, you are picking up random fragments. And in the process, you did not pick up fragment 9, 5, and 4, for example. So since these fragments 9, 5, and 4 have not been picked up for sequencing, obviously their sequence will be missing. So when you assemble the fragments together, you will not get sequences for 9, 5, and 4. You have one Contig here between 1, 2, and 3. You have a second Contig here between 6, 7, and 8. 4 and 5 are missing here. And then you have a third Contig here between 11 and 10. Fragment 9 is missing here. So these represent your gaps, and the continuous sequence that you get based on the overlap represents your Contigs. Gap can be of two types. One is the sequencing gap, where the fragment representing the part of that genome is present in a library. Only thing is that when you're randomly sequencing the fragments, you did not pick up these fragments for sequencing at all, right? So this is a sequencing gap. The fragment linking the Contigs is present in the library, but has not been sequenced, right? And second is what is known as a physical gap. And physical gap occurs because of the fact that some fragments are incompatible to certain vectors, and therefore they are never a part of your library. They are either not ligated or not transformed into the host cell. So in that case, what is happening here, if this was to represent a physical gap, then you will not have colonies 9, 5, and 4 in your plate at all, right? So that would be an incomplete genomic library. So the next idea then is to how to resolve a gap. So up till now, what you have done is you have made a genomic library, then you have sequenced individual fragments, and based on the overlap data, you have got your Contigs. The next challenge now is to finish the gaps or complete the gaps, right? So how do you go about completing the gaps is what we discuss next. But before that, let me ask you a question, right? And also give you an idea of nucleic acid hybridization, which would play a major role in resolving the gaps in your genome assembly. So first up is the question, and then the concept of nucleic acid hybridization. So here we are, and the question is, 150 contigs are found after assembling the Sanger reads obtained from a genome sequencing project. Estimate the minimum number of gaps that are present. And your options include A, 75, B, 149, C, 150, and D, 151. So to answer this, let me take you back to the previous slide. And if you see here, there are three contigs, right? So this is contig 1, contig 2, and contig 3. And clearly there are two gaps in between the contigs. So if you see here, there are three contigs that are given to you. And if you look at the internal gaps between the contigs, there are two gaps, one between contig 1 and contig 2, and one between contig 2 and contig 3, right? And if there were only two contigs, then you know there would have only been one gap. Had there been four, there would have been three gaps, which are internal to the contigs. You could also have gaps beyond this because you may not have sequenced the ends of your genome, right? So therefore, in the question, what is asked is the minimum number of gaps given a contig number. So given a contig number of n, the minimum number of gaps that you can have is n minus 1, which would be, for example, here in case of 3 contigs, it is 2. In case of 2 contigs, it is 1. Right? So here in this case, if you have 150 contigs, the minimum number of gaps that you would have is 149, right? So this is correct. 149 is correct. So now we come to the next concept here, and this is a concept that is universal and is used not just in genomic libraries, also for northern and southern experiments. So this is known as nucleic acid hybridization. So here you are, and hybridization is a phenomenon in which single-stranded nucleic acid molecule can find an allele to its complementary nucleic acid molecule. So hybridization is a phenomenon based on the double-stranded nature of DNA and the complementary nature of the two strands, right? So hybridization is a phenomenon in which single-stranded nucleic acid molecule can find an allele to its complementary nucleic acid molecule in the presence of a large number of other non-complementary molecules. So here I show you two DNA strands which are complementary to each other. So these two DNA strands, when they are found amongst a population of a large number of non-complementary strands, they would still be able to find each other and complement with each other and form a stable double-stranded DNA molecule based on the hydrogen bonding between complementary nucleotides. So adenine pairs with thymine with double hydrogen bonds, and guanine pairs with cytosine with triple hydrogen bonds, and that allows for two complementary nucleic acids to look for each other and also hybridize with each other amongst a complex population of other non-complementary DNAs. The other important thing here is that the two strands need not be of the same length. You could have one very long DNA strand and the other strand, which is complementary, could be very short, and it would still be able to scan through the longer DNA, find its region of complementarity, and hybridize by forming hydrogen bonds. And it's something that regularly happens when you do your polymerase chain reaction. The primer is just about 18 to 27 bases in length. And let's say that you're doing a PCR on human genomic DNA, so that would be around 3.2 billion base pairs, and still that 18 to 27 base pair single-stranded DNA or primer is able to find its region of complementarity and anneal to that region. So that is what is hybridization. So to check whether a specific fragment is present in a library or not, what you can do is you could use a complementary DNA sequence, for example, this one here or this one here. And the complementary DNA fragment is coupled to a reporter molecule, which could be a fluorochrome or a radio label, right? So either of the two can be used here. So now this complementary piece of DNA that you introduce into your library and check for hybridization is called as probe, right? So here you are, the single-stranded nucleic acid molecule carrying a label, radioactive or fluorescence, is called a probe. A probe is used to identify the fragment of interest from a library of fragments via southern hybridization or northern hybridization. So let's now look at the steps in a library screening procedure. So here is your library in a culture plate, and this contains now colonies that have individual fragments of DNA coming from your original genomic DNA that you had fragmented. So the first important step, of course, is that you have to transfer these libraries onto a synthetic membrane or to a cellulose pig. So the membrane into which you want to transfer your colonies is basically put on top of this Petri plate here. So once you are sure that the colonies are transferred onto the nylon membrane, the nylon membrane is taken off the master plate here. So here you are. And now if you see, the nylon membrane is a mirror image of the original plate here. And to ensure that you're able to locate the exact colony, what you do is you make your points of reference. So this one is point number 1, this one is point number 2, which is present in the nylon membrane here, and in the original plate it is present here. Remember, mirror images, right? And then this is your point number 3 here, and this is the reference number 4. So these presence of these marks allows you to exactly and unequivocally locate the colony that you're referring to in the nylon membrane with respect to the original master plate here. You could do a denaturation of DNA here. So let's see, in this nylon membrane now, I'm interested in finding the colony that contains my fragment 3. What I do is, again, a nucleic acid hybridization. I design a probe that is complementary to fragment 3 and carries a radio label along with it. And once I do my hybridization onto this nylon membrane here, only the fragment 3 would show up as a signal in my autoradiogram, right? So here you are. I can now reference it back to this third position here or the third position here in the main master plate library. So now what I've done is from this, I have now identified which colony contains my fragment 3. I can now pick up the colonies from here, do a mini prep and isolate the plasmid and sequence the plasmid to get the actual sequence of fragment 3 here. So in the context of genome sequencing, if you look at library screening, the library screening helps you identify the colony that contains a fragment of interest. So we now move on to the next part of the talk, and where we are going to talk about how to resolve gaps in the genome assembly. But before that, let's have a few quick questions. So here is the first question. Which of the following blotting techniques does not involve nucleic acid hybridization? And your options are A, northern, B, southern, C, western, and D, none of the above. Both northern and southern are examples of nucleic acid hybridization. Of course, the starting material is different in both. So in case of southern hybridization, the starting material is genomic DNA or DNA per se. In case of northern hybridization, the starting material is actually mRNA, which is converted to cDNA. And then, of course, you're looking for the expression levels of a specific gene in case of northern. In case of southern, you're looking for the presence of a specific fragment of DNA in your complex library here. Western, on the other hand, is basically you're looking for your protein of interest in a complex mixture of proteins by using an antibody against that protein of interest. So the correct answer for this question would be Western. So here's the question again. Which of the following techniques involves nucleic acid hybridization? And your options are A, northern, B, southern, C, CRISPR-Cas, and fourth option is all of the above, right? So as I told you in previous question, of course, northern and southern require the nucleic acid hybridization. So the only term where you might have a confusion is CRISPR-Cas, and I'm sure you would have heard this term CRISPR-Cas very often nowadays. This is a technique that is being used very extensively for genome editing, and now, of course, it is found used beyond genome editing into trying to introduce other changes into the genome, as also in diagnostics, right? So CRISPR essentially stands for cluster of regularly interspaced palindromic repeats. CRISPR is a gene editing system, and the location where the edit will be made is determined by the hybridization of a guide RNA onto the target DNA on the basis of base complementarity, right? So therefore, in this case, the correct option would be all of the above or D. Right? Jennifer Doudna and Emmanuel Charpentier were awarded 2020 Nobel Prize in Chemistry for their discovery and application-oriented research for CRISPR-Cas, right? So let's now get back to assembling the genome and resolving the gaps. So we talk about the last step in genome sequencing project that is known as genome finishing. And here in this case, we're trying to resolve the gaps that have been noticed after the alignment of the reads that we obtained. So first of all, we would like to resolve the sequencing gaps. A sequencing gap essentially means that your library was complete. However, because you are picking up fragments at random for sequencing, some of these fragments are not picked up and therefore they appear as gaps in between the contigs. So the gap could be small in size or it could be very large in size, right? So depending on how big or how small a gap is, there could be one or multiple different fragments in between the gap, right? So the sequencing gap could be small or large in size. By small, it means that a single fragment is able to join the remaining contigs together. So for example, here, this is your full genome here, and you have 1, 2, 3 forming contig 1. Likewise, you have 5, 6, 7, 8 forming contig 2. However, the colony that contained the fourth fragment has not been sequenced. And because it has not been sequenced, you have a gap here. And therefore, from the master plate, you have to isolate the colony that contains fragment 4. So the gap could also be larger in size, and which essentially means that more than one single fragment is present in the gap. So here is an example of that scenario. So you have these 1, 2, 3, and based on our lab data, they form contig 1. Likewise, 5, 6, 7, 8, based on contig data, they form the contig 2. However, in between contig 1 and contig 2, there are actually 3 different fragments, A, B, and C, which need to be sequenced before the gap can be bridged. So the job basically is to identify A first, sequence it. Based on this sequence, design a probe to identify colonies containing the B fragment, and then of course to identify the colonies carrying the C fragment, and thus allow the assembly of the genome. So let's start with the first example first, where the gap is small and is likely to be covered by a single fragment, right? So here you are, this is your genome, and you have 2 contigs here, contig 1, which is basically made by joining reads 1, 2, and 3. Likewise, you have contig 2. And then we have fragment 4 that can connect contig 1 to contig 2. However, we have not sequenced this, and therefore this is a sequencing gap. So the first job in the master plate is to identify which of these colonies contains the fragment 4. Right. So here is our master plate, and amongst the colonies that are there here, our challenge is to identify colony number 4, which contains the fragment 4. So what we do here for library screening is to develop 2 probes. One of the probes is towards the 3' end of the third fragment or towards the end of contig 1. Likewise, you have a second probe that is developed towards the 5' end of contig 2, right? So when P1 is used as a probe, it will obviously go and anneal to 3 because it is being designed from there. Plus also it will anneal to 4, to which it has a common overlap. So when you probe your library with P1, then you will get a signal, of course, at the colony that contains fragment 3. Also, you'll get a signal in the colony that contains your fragment 4, right? And you already know the location of fragment 3. So the other colony that gives you the signal would be your actual colony, from where you can pull out the plasmid and sequence it to get the sequence of fragment 4, right? So when you probe your library with probe P2, you get 2 signals, one for fragment 5, from where you have designed the probe, and second for fragment 4, to which it shares a common end. So when you probe the library with probe 1 and probe 2, you get one common colony, which is highlighted in both the library screenings, that is colony 4, which indicates that there is a single fragment that is able to bridge the gap between contig 1 and contig 2 and give you a continuous sequence. Right. Another strategy to identify the fragment that connects contig 1 to contig 2 could be to use what is known as colony PCR, right? So you basically use primers P1 and P2 to amplify a piece of DNA coming from these colonies here, right? So in case of 2, both P1 and P2 will not bind, so no amplification. In case of 7, again, both P1 and P2 will not bind, no amplification. In case of fragment 3, P1 would bind, but P2 will not bind, therefore no amplification. Likewise, fragment 6, neither P1 nor P2 would bind. So therefore, the only place where you can have amplification using primers P1 and P2 is going to be fragment 4 because it has annealing sites for both P1 primer and P2 primer here. So this could be your alternate method of identifying the colony that contains fragment 4, and this is known as the colony PCR method. So a colony PCR is just like a normal PCR. It will have all the important components of a PCR, pairs of primers, DNTPs, DNA polymerase, magnesium chloride, the buffer, as also the water to make up the volume. And the DNA is added indirectly by pricking the colonies with toothpicks and then releasing the contents of the tip into the PCR mix. However, there could also be cases where the gap is large and more than one fragments are involved in between. So here in this case, for example, you have the genome again, and then you have the first 20 consisting of the first three reads, 1, 2, and 3. Likewise, you have the second contig that is again consisting of reads 5, 6, 7, and 8. This is called contig 2. However, in between, there are more than one fragments that are present. So now your job here is to identify the first fragment in the order. Once you identify the first fragment in the order, sequence it, identify the second fragment in the order, and then again sequence it, and then design primers to identify the third fragment in that order, right? So here is your genome. These are your Sanger reads, and based on the unique overlap data, you combine 1, 2, and 3 together to form Contig 1. Likewise, reads 5, 6, 7, and 8 can be combined together to give you Contig 2. And in between Contig 1 and Contig 2, there are 3 different fragments that are present. So in this case, you identify the fragments in a sequential order. First, you identify A. So in this case here, Contig 1 and Contig 2 can be basically bridged only when you have the sequence of A, B, and C all put together, right? So what you do here is in this case, you basically go sequentially. You first use a probe against the 3' end of fragment 3, which will then be used to identify the colony A here, right? So it will then be used to identify the colony A here. So in this case, when you use probe P1, which is basically having sequence commonality with 3' end of fragment 3 and also the 5' end of fragment A. So this probe is going to bind to both 3 and A. You already have the sequence of 3 and you know what is the location of 3. The other colony to which this probe binds will give you the idea of what is the next fragment in order. So to resolve the gap containing 3 reads A, B, and C, first you screen the library with probe P1, which is complementary to the end of the Contig 1 or clone 3, this one here, and it also has overlap with fragment A. So this will go to 2 positions. The probe shows signals in clone 3 and in clone A as well, which overlaps with clone 3. So now you have identified A, you sequence A, and now design a probe against the 3' end of A, and use the same strategy to identify the colony containing the B fragment. Once the B fragment is identified, again, design a primer to the end of B and use that to identify the colony that contains fragment C in your master plate and sequence that as well. Once you have sequenced A, B, and C together, you will be able to assemble the entire genome based on the overlap data between the contigs. So once you have sequenced A, B, and C, you will be able to bridge the gap between Contig 1 and Contig 2. So here you are, you sequence A. After you've sequenced A, you repeat the experiment to identify B, and then you repeat the experiment again to identify C. You keep repeating this until the gap is sealed.